for the continuation of our topic. So, photosynthesis process is divided into two major steps, that is light reactions and dark reactions. Light reactions takes place in the grana, that is thylakoids membrane, and light reaction is divided into steps. There are absorption of light and splitting of water molecule, then formation of assimilatory powers, that is ATP and NADPH2. Before going to start the light reaction, we must once again we will recollect the, if you recollect the structure of, that is chloroplast. So this is the chloroplast and thylakoid. So in this thylakoid membrane, it is the single thylakoid. In this thylakoid membrane, our special pigments, that is chlorophyll pigments and other pigments, are associated in the groups. So that groups are known as quantasomes. Quantasome or photosynthetic unit. What is that? Photosynthetic unit. So we know that the chlorophyll pigment, not only chlorophyll pigment, carotenoids, xanthophylls, they are present in the thylakoid membrane. So all these pigments, they are group of pigments collectively called as quantasome. What is quantasome? Quantasome is a collection of pigments. Collection of pigments. Collection of pigments. We can simply say that it is a collection of pigment. So what are the pigments? The main pigment is chlorophyll A. That is, main pigment is chlorophyll A, and it is associated with, it is gathered with other pigments. So this is known as reaction center molecule, and other molecules are known as accessory pigments. All this structure, this is group of, what is this group of? All these are the pigments. Other pigments, what are they? Xanthophylls, carotenoids, all the pigments are gathered, collectively present in the thylakoid membranes to capture the light, to absorb the light. That is called as photosynthetic unit or quantasome. Nearly how many pigments? Nearly 200 to 450 pigments are gathered in that. They are capturing light. They are acting as an antenna. It is also called as antenna complex. What is the complex? Antenna complex. So in this excited chlorophyll, so there is a special definition is given to the quantasome by the Emerson and Arnold. Emerson and Arnold. What they are giving, that photosynthetic unit as the minimum number of chlorophyll molecule, capable of acting collectively in the photophosphorylation to evolve one molecule of oxygen. I will write it here. So what they are, that is, so photosynthetic unit as a minimum, it is photosynthetic unit as a minimum number of chlorophyll molecule, capable of acting collectively. How they act? Collectively they gather. Capable of collectively in photophosphorylation. In photophosphorylation to form how many molecules? One molecules. To evolve, to release or evolve one molecule, one molecule of oxygen. Once again, Nana. So, what is the definition of this quantazo? Photosynthetic unit. This is the photosynthetic unit as a minimum number of chlorophyll molecule. Here the minimum number of chlorophyll molecule should present and capable of acting collectively. There not only single chlorophyll molecule is not there in the quantazone. It is a collectively some group of pigments are gathered. They are acting like an antenna to capture the light in the form of photons. Collectively photophosphorylation. Photo means light, phosphorylation means synthesis of ATP and NADPH2. So they are collectively acting to synthesize the assimilatory powers and to evolve the one molecule of oxygen. We know that the byproduct of photosynthesis is oxygen gas is released. That is produced in the phosphorylation only in lumen of thylakoid. So to produce one molecule of oxygen, how much of elements or how much of pigments along with the chlorophyll pigment are required? That is known as a photosynthetic unit or quantazone. This definition is given by Emerson and Arnold. So what is the photosynthetic unit? So some group of pigments, they are gathered to capture the light. That complex is known as photosynthetic unit. In the photosynthetic unit, our main pigment chlorophyll A is act as a reaction center and other pigments act as a accessory pigments. What happens when the sunlight falls on all the pigments, they capture the light and they pass to the reaction center. And by taking the light energy, it is going to be activated and release the pair of electrons. Those electrons are accepted by primary electron acceptor. They are accepted by primary electron acceptor and it enters into the electron transport chain and finally it produces the ATP and NADPH2. So this is the quantazone. Quantazone is the reaction center. This type of quantazones are present in the thylakoid membranes. They are present in the thylakoid membranes. So these quantazones, they are present in the form of specialized systems. To capture the light, there are two special systems. Special systems are present in the plants, that is known as photosystem 1, photosystem 2. So what are these photosystem 1 and photosystem 2? We will write them as photo PS1, PS2 and also written as P68, P700 nanometers and P680 nanometers. Why we will write that? So in the plants to capture the light, there are special pigments are arranged in the form of photosynthetic units. So these photosynthetic units together to capture the light, there are two types of photosystems are present in the plants. They are known as photosystem 1 and photosystem 2. So if you say the reaction center, same in the photosystem also there is a reaction molecule, reaction center, chlorophyll A molecule, it is surrounded by other accessory pigments, and so this is chlorophyll A and accessory pigments. This is reaction center. In each and every photosystem there will be reaction center and accessory pigment and PEA, primary electron acceptor. Primary electron acceptor. So when excited electron, when it absorbs the light, when chlorophyll molecule and other pigments, they absorb the different wavelengths of light. So to absorb the light, all the pigments behave independently. Independently they absorb different wavelengths of light. So they absorb different wavelengths and they pass to the reaction center, that chlorophyll A molecule, and by taking the high energy, the outer it is going to release the electrons, pair of electrons. So it releases the pair of electrons. They are accepted by a complex that is known as primary electron acceptor. Which one is going to accept these elements, these electrons, that is known as primary electron acceptor. In the case of photosystem 1, so reaction center, this chlorophyll A molecule activate at 700 nanometers. So 700 nanometers. So this system is also known as P700. P700. Okay, photosystem 1 can be written as a PS1 or P700. In these photosystems, what we have, we are having the components. One is reaction center, that is, it is also known as an antenna complex. In the antenna complex, we have the reaction center molecule and other accessory pigments. So this is the photosystem. There is a major, what is the major difference in between the photosystem 1 and photosystem 2? That is the wavelength of light, what they will take. In the photosystem 2 also, there is a reaction center molecule, chlorophyll A, and it is associated with the other accessory pigments. But the wavelength which it is accept is 680 nanometers. So same thing happens. By the absorption of light, all the pigments, they absorb the light and they pass to the reaction center. It is activated and it release the pair of electrons. Those pair of electrons are accepted by the primary electron acceptor only. Primary electron acceptor. So what is happening here? So where these systems are present. So this is the chloroplast. So in chloroplast, one thylakoid we are taking. Thylakoids they are connected with the what? Stoma lamellae. In this thylakoid membrane, PS1, PS2, PS1 is present. But whereas in the lumen of thylakoid, only PS1 is present. PS1 is present in the stoma lamellae, whereas lumen of membrane of thylakoid, in the membranes of thylakoid, PS2 and PS1 are both are present. So they activate, they synthesize in non-cyclic photophosphorylation, they synthesize the ATP and ADPH2, whereas in the photosystem 1, in the cyclic photophosphorylation, they synthesize only ATP molecules. So where these systems are present? So photosystem 2 is present in the thylakoid lumen, but whereas in the photosystem 1, it is present in the thylakoid membrane as well as the stoma lamellae. Both the sides it is present. Okay, so it is present in the both sides. What are the photosystems? So in green plants, to capture the light, to gather the light, there are two systems, two antennas. We can simply say that two antennas are there. So two pigment systems. We can also call them as a pigment systems. Two pigment systems. Two pigment systems are there. One is photosystem 1 and photosystem 2. So they are named photosystem 1 and 2 by the discovery. First it is discovered PS1 and then it is PS2. So the absorption wavelength is photosystem 1 activates at 700 nanometers, whereas photosystem 2 activated at 680 nanometers. This is the information about photosystems. Photosystems are present in these systems are present inside the chlorophyll. Photosystems are nothing but group of pigments, group of chlorophyll pigment along with the other accessory pigments. They are acting like an antenna to capture the light. Okay. So this is also known as light harvesting systems also. They are also known as LHC. L means light harvesting complex. Light harvesting complex. Light harvesting. Why we are saying this is a light harvesting complex? Because all the pigments, they are capturing different types of wavelengths of light for the process of photosynthesis. For the process of photosynthesis, they are capturing different wavelengths of light. Light may be having different wavelengths. So if only one pigment is present, so there is a rate of photosynthesis may be decreased. So that is the reason all the pigments, they gather together and they capture the different wavelengths. That systems are arranged in a two groups, that is photosystem 1 and photosystem 2. They act as a antenna to capture the light. Okay. So light dependent reactions. So next, coming to the light reactions. Light reactions, first process of photosynthesis is divided into light reactions and dark reactions. In the light reactions, we have the light absorption, splitting of water molecule, and next formation of assimilatory powers. So activation, that is light absorption or activation of chlorophyll is also required the light, whereas splitting of water molecule also it is depends on the light only. And then finally assimilatory powers, that is ATP and NADPH2 are produced. So all these reactions are depend on the light. So that reactions are known as light dependent reactions. Where they take place? Where the photosystems are present. Where the photosystems are present? In the thylakoid membranes. So site of the light reaction is thylakoid strictly. That is thylakoid. So this light reactions are first light absorption, that is photophosphorylation is two types, that is non-cyclic and cyclic photophosphorylation. Once again all the recapitulate all the things. So quantazome or photosynthetic unit, quantazome or photosynthetic unit is a, so those are present in the thylakoid membranes to capture the light. So it will have the one chlorophyll molecule, at least one chlorophyll molecule and other accessory pigments. So they take the light and they release the electron, excited electron, that is accepted by the primary electron acceptor. So one more thing, that if we isolate the chlorophyll, only if we are isolating the isolated chlorophyll. We are isolating the chlorophyll, we are taking the chlorophyll and isolated, we are taken in the test tube and exposed to light. What happens? It is in the closed condition, experimental condition. In the experimental condition, there is no primary electron acceptor. So it releases the excited electron, and this excited electron with the high level of energy, when it is excited, it will have the high level of energy, and there is no primary electron acceptor. To take this electron, there is no complex is not there. So ultimately, it comes back to the reaction center only. It comes back to the chlorophyll pigment. While it is coming back, it releases energy in the form of light. So it releases the energy in the form of light. So this process of is known as fluorescence. Fluorescence. What is the meaning of fluorescence? That in the experimental condition, when we isolate the chlorophyll molecule and when we expose to the light, what happens? So the excited electrons will come outside and again they reach to the chlorophyll molecule only. So while they are coming down to the energy level, they release the energy in the form of light. That process is known as fluorescence. Okay. In the next we will see about the non-cyclic and cyclic photophosphorylations.